Thank you. I'm Nichole Reisdorph. I direct the Mass Spec Core over in the School of Pharmacy, but don't let that fool you. We are actually a campus resource, not just for School of Pharmacy. And we have collaborators all over the United States. So I will be presenting our basic services, but really trying to highlight some of the aspects that make our core a little bit unique. Make it a little bit unique in how some of the services we offer can hopefully help enable some of your research. So for a quick overview, we are really a one-stop shop for mass spectrometry. We have special skills in metabolomics, lipidomics, foodomics, also in proteomics. And then one of the specialties of our core is that we have what are called targeted quantitative assays. And so we can quantitate over 250 small molecules and peptides using one of 15 different assays. So whereas metabolomics and proteomics are more discovery-based research, we can also help you with more targeted quantitative information on specific molecules, and I'll give you more detail on that coming up. We also conduct workshops. These are 4-day hands-on workshops. We've been doing this for well over 15 years now. And in addition, our specialties in terms of research focus on respiratory diseases and also foodomics and Nutrimetabolomics. And I'll give you a little bit more information on those too. So that's just a snapshot. I'll go into the individual sections that we have: proteomics, metabolomics, and targeted small molecule assays next. So in terms of proteomics, we can do all of the basic proteomic assays and experiments that you need. We have specialties in acelomics or acetylomics, and also a quantitative proteomics workflow that is label-free. So this label-free workflow can help with projects that require some increased sensitivity. We do extensive fractionation so that we can really dig deep into individual proteomes. And once we do that and identify as many proteins as possible within your system, then we go back and do a label-free type of quantitation. This is called accurate mass retention time library for those who are familiar with proteomics workflows. So any basic proteomic experiments too. Our laboratory has really started focusing and specializing in something called acelomics. And so this is site-specific post-translational modifications on that result in acetylation, succinylation, malinylation, and propinylation. Cole Mitchell in our lab worked very closely with Chris Fritz at School of Pharmacy and David Ross and Manisha Patel in really developing these targeted assays. They can be performed in any number of sample types, including skeletal muscle, heart, brain, liver, eye tissues, and Cole can also do subcellular fractionations. This can be combined with quantitative proteomics to give you really good information on whole protein changes, as well as in the site-specific modifications. The figure that you're seeing in the bottom right was put together by Cole for a collaborator, Doug Lewandowski, in Illinois. Hopefully this publication will be coming out soon, but it can really show that you can focus on very specific proteins and very specific sites and get an idea of energy metabolism within the cell. And in this case, I think he's looking at the mitochondria. Cole has several publications, and the lab has several publications in this area. I'm including different citations. If you want a copy of this presentation, just email me and I can give you a PDF, and always happy to send proteomic citations. I put this up here also just to show you that we have a variety of projects in proteomics, not just acelomics or quantitative proteomics, but also working with our various collaborators on identifying novel peptides, novel proteins in their specific disease states. And then we can also take everything that we've discovered using discovery-based proteomics and turn it into a targeted assay so that you can get absolute quantitative information on specific peptides, for example. In terms of small molecule analyses, our laboratory has extensive experience in metabolomics, and I want to use this opportunity to first really distinguish between the types of small molecule analyses and the types of mass spectrometry in general. So I think most people are familiar with metabolomics, proteomics, maybe even lipidomics. These are discovery-based assays where you're trying to generate hypotheses. You don't necessarily have set hypotheses about your specific system. This is also called biomarker discovery. So I'm trying to find new small molecule or protein biomarkers. Some people refer to this as metabolite profiling. There's also targeted assays. These take place on completely different sets of instrumentation. The technologies are different. So for discovery-based metabolomics, this results in relative quantitation. So for those of us on the call, we can divide maybe into males and females and say that the males have higher levels of X metabolite compared to the females, or those of us with short hair have more of one metabolite. These are known molecules, so it might be testosterone or vitamin D, for example. So if you go to the doctor and they order a vitamin D test, you're going to get a result in, say, picograms per mil of plasma. And that's the sort of information we'll get with a targeted assay. We can give you picograms per mil. So these are more appropriate when you're conducting basic research or clinical studies that span over, say, weeks to months, where we can't run all of the samples in a single batch. And that's more important for metabolomics, discovery based. We can still do cross-study comparisons, just not quite as easily. The other distinction is this term lipidomics, and that's really trying to get higher coverage of lipid molecules. And we can do lipidomics in a discovery-based mode where we're enriching for lipids and just doing hundreds to thousands of comparisons of lipids. Or some people are referring to lipidomics in more of a targeted sense. So we do have targeted assays that focus on specific lipid classes, such as the oxylipins. These are also called lipid mediators, and I've got an example of that coming up. We can also kind of do a hybrid whereby you're looking at more of a general pathway. So you just want to see the aqueous metabolites compared to the lipids. So hopefully that clarifies a little bit. Our laboratory does both of these, and the workflow that we generally use for a new project that is trying to do discovery-based metabolomics or proteomics is to use those technologies to discover some pathways of interest or molecules of interest, and then we can go back and help you develop a method that's specific for certain pathways, or maybe we already have a method existing to help you really interrogate that pathway in a little bit more depth. So we have, I have over 20 years of experience in metabolomics. If I try and add up all the years of experience in the laboratory, we get to an obscene number pretty quickly that might be showing our ages. So suffice to say, we have a lot of experience in metabolomics, proteomics, and mass spec just in general. For metabolomics, a lot of people again use this for biomarker discovery. You might want to look at a specific pathway. We have experience in any biofluid I think you can think of, cells, tissues, nanoparticles. Metabolomics is appropriate for clinical studies, for mouse studies, any type of translational research. So it's really a kind of a general purpose We know that our metabolomics will give you coverage of the molecules that you need. However, a lot of people are using metabolomics for hypothesis generation. If you do have a specific hypothesis you're trying to test, usually we have a targeted mass spec assay that can fill that need. But if you're trying to generate new hypotheses, metabolomics might be the way to go. If you want really broad coverage, I want to look at, as many possible pathways that I can, metabolomics might be a good choice for you. And again, looking at a broad range of chemical classes from lipids and amino acids. So these are the endogenous molecules. We also measure a lot of exogenous molecules in metabolomics, a little bit different from the other ohms that way. So anything you ate for breakfast, any exposures that you had today, if you, maybe not this morning, but if you ran to work or rode your bike to work and got behind a bus, you've probably got some pollutants in your system, and metabolomics can measure some of those as well, as and our targeted assays can measure some of those as well. So again, the workflow that we usually use is metabolomics for discovery purposes, and then we go to more targeted approaches. Newest in our laboratory is a sample preparation robot. We're currently testing this for metabolomics, but it is able to be used in proteomics and small molecule analysis too. Mass spectrometry is extremely expensive, including service contracts, and we try and keep our prices low, but the equipment itself is very expensive, and the expertise, of course, is expensive. So we brought this robot in hopes of decreasing prices and easing the strain on people's hands for pipetting 300 or plus samples. We're starting to run a lot more very large projects now, and so this robot hopefully will come in handy. That wasn't supposed to have sound. Sorry. So this is just the robot in action. I don't know if you guys can see that working or not, but it's meant to be a cost-effective solution for sample preparation for the laboratory. An example of what our laboratory specializes in terms of metabolomics also really highlights some of our skill set expertise and what metabolomics in our lab can do for you. Nutrimetabolomics is the intersection of clinical nutrition research and metabolomics. And of course, this campus has a very, very strong program in nutrition and clinical nutrition. So we're using metabolomics of both foods and biospecimens. So we do metabolomics on our library is now well over 100 different foods. We'll use urine and plasma from study participants, and we can use this information in a lot of different ways. So for example, looking at what specific compounds within a food are responsible for health outcomes. And you can take that a step further and say what compound within blueberry is actually helping decrease blood pressure. And so we have NIH funding to perform studies like that. We can also look at what rate compounds are metabolized. So we have several acute feeding studies where individuals are given a meal. We do metabolomics of the foods within the meal, and then also metabolomics of the plasma samples across, say, up to 10 hours. And then we can look at the food compounds and see how quickly or slowly they're metabolized. Do they show up at all? Asking questions like that. Nutrimetabolomics is also being used to determine biomarkers of dietary intake. And we don't have grants that focus specifically on that, but sort of incidental to determining what foods, what compounds are specific to certain foods, we can also find biomarkers of intake. So if you're in a clinical trial and you're supposed to be eating a lot of blueberries or a lot of bananas, we can find markers of blueberry in your plasma or in your urine. Don't be scared, we're not actually doing this right now. And determine if you're staying true to that dietary intervention. Another way that we can use nutrimetabolomics is to determine what foods or cultivars of foods contain higher or lower amounts of specific compounds. And we do this using targeted mass spec. The ones above we're using predominantly metabolomics, so discovery based. But we have found, for example, that astaxanthin is higher in wild versus farm salmon. And so our collaborators have studies looking at giving individuals wild versus farm salmon and looking at health outcomes, both at the Anschutz campus and at Children's Hospital. Another example, red bell pepper has more cryptoxanthin. And so to understand that level, we do targeted mass spec. We develop assays for astaxanthin, cryptoxanthin, other polyphenols. other xanthan molecules, and we can measure absolute amounts. So for example, picogram per milligram of food. I think this might take a little bit too long to go through, but suffice to say that we do metabolomics of the foods, and that's shown up top. We do extensive annotation, extensive efforts to try and identify as many molecules in food as we can. We currently, our food library is building right now, our food compound library. We should be receiving soon another 1,000 compounds. So we'll be able to help identify in the course of the next 6 months, about 1,500 food compounds, actually know what they are instead of just giving them an annotation. Metabolomics is notorious for we can't really identify molecules really well, but with these food compound libraries, we'll be able to identify about 1,500 food compounds. Our plasma and endogenous molecule library is over 600 compounds right now. So we determine the compounds in food. We can also look at plasma and urine before and after a dietary intervention, and then we can look in those biosamples for these food compounds, and then we can associate those with health outcomes such as blood pressure. I can't seem to get my, there we go, such as blood pressure. These are just a few of our metabolomics citations highlighting our Nutrimetabolomics. We've also got studies focused on insufficient sleep. We have a nice program building with Kathy Lozapone and Dan Frank on campus, integrating microbiome and metabolome data sets. As highlighting some of our respiratory research, we've got a lot of publications in COPD and asthma that focus on both metabolomics and targeted assays. We've done nutrition studies. It's not really nutrition, but basic research in mice or in rats looking at ketogenic diet. And then we have quite a few more technical papers to just developing methods, optimizing methods, and just trying to improve the field in general. So again, in terms of lipidomics, we can do both kind of a large scale comparison of many different classes of lipids, including all of the glycerophospholipids, sphingolipids, et cetera. And then we can do targeted lipidomics assays, and I'll give you an example of that coming up. Lipidomics is becoming really more of a distinct field compared to metabolomics, which focuses more on the aqueous or hydrophilic molecules. So again, in terms of metabolomics, you can use that for discovery. If you have specific hypotheses to test, we may recommend that you use one of these targeted mass spectrometry assays. This is also called absolute quantitation instead of relative quantitation. And we have about 15 assays that cover well over 260 molecules. And these are performed by LC, liquid chromatography mass spec, and also GC, or gas chromatography mass spec. And so overall, metabolomics will give you a snapshot of many different pathways, whereas these will help you drill down into specific pathways. I'm not expecting you to read through all of these, but as some examples, we can measure acylcarnitines, bile acids, endocannabinoids, neurotransmitters, short chain fatty acids, hormones, TCA cycle intermediates, and then you can narrow in on any of these. So if, for example, you're interested in inflammation, there are a lot of small molecules, lipids in particular, that are also good readouts of inflammation. So you may choose to measure oxylipins, and some of these are pro-inflammatory biomarkers such as leukotrienes, prostaglandins, isoprostanes, for example. Some of these are pro-resolving, so they would be at a higher level when someone is getting over inflammation, for example, like I am. You might hear it in my lungs. So these are like the maricins and resolvins, protectins, for example. And so if inflammation is something you're interested in, we may recommend that you look at both the precursors, arachidonic acid, DHA, EPA, other fatty acids, and then look at these pro-inflammatory and pro-resolving lipids. If you're interested in gut microbiome, we may recommend that you look at short-chain fatty acids in the feces, perhaps in the plasma. And then maybe if you're interested in serotonin, which is largely produced by the microbiome, you might be interested in looking at tryptophan metabolism as well. So depending on what your specific focus is, we can help you kind of tailor these targeted assays to help you answer some specific questions and really dig in and look at how certain molecules are changing. This is just showing just a snapshot of the oxylipin assay where we're measuring over 90 oxylipins. PUFA is polyunsaturated fatty acid metabolism. So the precursors, arachidonic acid, DHA, EPA, these are the omega-6s and the omega-3s listed on top here. And then down here, giving rise to all of these lipid mediators. The ones in red are the pro-inflammatory, the ones in blue are the pro-resolving. We've used this in a variety of research projects spanning arthritis, immune, obesity, and asthma, for example. So you're getting a lot of quantitative information on each of these individual molecules, and that allows you to look at ratios and changes depending if you're doing cells or tissues or plasma sample. You can look at changes due to various interventions. Another example is the NADome, or looking at the NAD pathway. This is another absolute quantitative assay. We did this one, I want to highlight this one in collaboration with Doug Seal's group at CU Boulder. And this is an example. We've done metabolomics with Doug's group before, but they really needed information on this pathway and specific molecules within this pathway. So Mike in the lab, Kevin also helped, and some previous lab members helped develop this assay to look at the NADome. And this assay is now being used to look at cellular metabolism, supplement use, whereby you've probably all seen like NMN, NAD being sold as supplements. And so we can measure these in a variety of biosamples, cultured cells, PBMCs, tissues, whole blood, etc. So this is the sort of service that we offer, where we help you develop the technology to enable you to make the measurements that that you need, and then we apply it to clinical studies. So we have one ongoing clinical study with Doug and Chris and their groups. Just a few quantitative mass spec citations, a lot of oxylipins on here, NAD. We also look at exogenous molecules such as polycyclic aromatic hydrocarbons. We can look at any number of drugs or drug metabolites using this quantitative mass spec as well. Nanoparticle formulation, really anytime you've got a molecule of interest that you think mass spec is a good way of measuring that molecule, we might be able to help you. So just to kind of reiterate, we do a lot of method development for people. And so we do a lot of collaborative discussions, sometimes taking days to weeks to months to help you guys build your project, help you get your grants, and then going in and actually developing the assays that you need to conduct your research. So anything from foods, drugs, peptides, intact proteins, polymers, lipids. We also do accurate mass determination for more of the chemists on campus. I think we have dealt with, I'm sure there's sample types we haven't dealt with, but it feels like we've dealt with almost every sample type. You can do metabolomics of joint fluid and bones, for example. We've done metabolomics and proteomics of mummies, CSF, feces, brain, sections of brain, lavage fluid from the lung, sputum, anything. So sometimes that goes into method development. If we've never worked with that particular sample type before, then we might have to optimize our assay for that particular sample type. But again, that's one of the things we specialize in. We also do GLP type and CLIA type method development for our targeted assays, so that you know you're getting really quality results back. I'm going to skip over this slide just to say that exposomics is another new field that's coming out, and we have all of the technology and a lot of the assays that you need to look at different exposures, environmental exposures, anything from foods to pollutants, pesticides, and that sort of thing. Overall, we love to be involved in collaborative research, and so I'm just highlighting some of the mutual funding that we've gotten recently from various collaborators, our Foodomics and Nutrimetabolomics. I'll show you some of our cannabis work where we're doing behavioral studies and looking at effects on PTSD and neurotransmitters. I mentioned previously the microbiome research that we're doing with Kathy and Dan. And we're also doing microbial metabolism with Christy Kuhn, Kathy Lozapone, Vanessa Fellon. We have large-scale projects in asthma, arthritis, a little bit in IBD now, various funding mechanisms, nanotechnology. We do a lot of we don't do a lot of metabolism studies. We do some very specific metabolism studies. So if you're interested in C13 labeling experiments, that's something that Mike and I have been doing for quite some time. They're very specific and very boutique. And so you want to really talk with us or Angelo and the other core about these type of C13 labeling experiments. These are completely separate from metabolomics. These are for very specific flux and small molecules. Some other collaborative research listed here, and I'm sorry if you're on the call and I forgot you. A lot of collaborative research going on. We also have industry partnerships. So just to give you an example of how the core might be able to help with a research project, we wanted to understand the effects of cannabis or other natural products such as green tea on mood-related disorders such as depression, anxiety, or PTSD. And so we're essentially going to be have fed mice cannabis or green tea, and we measure their behavior. So what molecules do you need to measure? It's one thing just to measure the mouse behavior, but what would you need to measure to really make this research kind of shine? Well, we developed a targeted mass spec assay to measure THC, CBD, and other cannabis metabolites in the plant to help determine how much of the plant do I give, because I don't trust NIDA when they tell me that there's this much THC in a sample. Also wanted to measure to determine what compounds are detectable in brain and how much, and then in plasma, what are the relative levels? And so we use targeted mass spec for those purposes. We also use targeted mass spec to measure neurotransmitters and endocannabinoids, which are directly related to mood. And then we use a discovery-based approach to discover new molecules or pathways of interest. And then metabolomics of the plant itself to look at new novel compounds that may associate with consumption or give us some hints about effects on mood. And then follow-up studies, we can use targeted mass spec to determine effects on inflammatory molecules. So you guys have the ideas, and then we help you measure the molecules of interest. We've done this in a cannabis experiment, and suffice to say, a lot of mood-altering neurotransmitters go up while endocannabinoids go down. And I know I have to wrap up here. So major equipment in the laboratory, we have an Orbitrap Eclipse with ETD scheduled to be delivered in January. We also have 2 QTOF instruments that are dedicated to proteomics. We have 2 instruments dedicated to metabolomics and use those other 2, the Eclipse and the ion mobility in conjunction with proteomics. For targeted assays, we have 4 different triple quads that we're using for those, and we also have an Agilent GCMS. As you can see, we have a partnership with Agilent Technologies. There's an ICP in the lab, standalone HPLC for fractionation, and then a mesoscale multiplex antibody system. We also offer training, as I mentioned. These are 4-day workshops. Hopefully these are going to start again next fall, post-COVID. Really, the #1 reason that you might want to use our core is the people. You really get to work with some exceptional people, many, many, many years of experience. Mike has, I think we're going on 30 years of experience. He and I have worked together for almost 18 years now. just a lot of good people, and they really care about your research and helping get your research to the next level. I'll end by just giving you some email addresses. Again, you've got my name on the screen. Feel free to contact me if you want a copy of these slides, and contact anyone listed here on the screen by email, or our phone numbers there too. Our website is a little outdated, so we've been working on that. Hopefully that'll be up again soon. So I'm happy to take any questions in the 1 second that's left. I don't see any questions, so hopefully you have the information you need.